right? And so we had talked about some different kinds of mutations, right? We talked about point mutations, right? Where just a single base gets changed, gets added, deleted, or swapped out for something else, right? With kind of a more specific example of a base substitution, right? That was the sickle cell anemia example where an adenine was switched out for a thymine, right? So you're switching out just one nucleotide base in the DNA code. It has some effect on the proteins that you make. And so these point mutations, we can kind of lump them into a few different kinds, right? So if we add, delete, or change, right, some base, right, it's possible because the genetic code is degenerate, right? You've got multiple codons, multiple three little combinations of nucleotides coding for the same amino acid, that you change one of those nucleotides, but you still make the same amino acid. And if you make the same amino acid, you make the same protein. So you could mutate the DNA potentially and have no actual downstream effect. And so a mutation like that is called silent, right? Because the mutation is silent. It doesn't cause anything different to happen in the way you're expressing your genes, right? Conversely, like the sickle cell example, you could change a nucleotide and it changed basically the meaning of the codon so that that codon now codes for a different amino acid, right? And that's called a missense mutation, right? Because now we've changed what the genetic code means. So we're going to put in a different amino acid, we're going to make a different protein, and then some series of effects will happen, right? And we lumped these into two groups, right? Because we've got four nucleotides, A, T, C, and G. Right, T and C are both pyrimidines, so kind of molecularly they're structured the same. And then the same for the purines, adenine and guanine, right? So if we switch out a pyrimidine for a pyrimidine, right, where it kind of has like, you can think of it as having kind of a lesser effect, right? Because thymine and cytosine kind of look the same. They're structured kind of the same. So it's a less intense sort of a change, right? Same thing if you switch adenine for guanine. But you can also have a transversion type mutation where you switch a pyrimidine for a purine, right? Which kind of molecularly is a more drastic change, right? And this, a transversion, is what is happening in that sickle cell situation, right? Because we switch out an adenine for a thymine. And then you can have nonsense mutations where you switch out one of those nucleotides and you turn that codon into a stop codon, right? Which is really problematic because the whole point is you've got this long string of nucleotides that code for these amino acids that make a protein. And if something in the middle gets turned into a stop codon, when that protein starts to get translated, it's just going to stop partway through, right? So it's nonsense mutation. You won't even complete the protein if you have that kind of a mutation. And so we can kind of see that happen, right? So in a missense, right? So we switch something in this arginine here, right? This is an AT situation. We switch that out and that arginine becomes a histine. And so now we've kind of changed the meaning of the code. So we've got a missense mutation. You can have silent mutations frequently in that third nucleotide position in the codon. That's where codons are a little less picky as far as what amino acid they code for. So we had a proline, we switched CG for this AT, but it still codes for proline. So the actual protein we make doesn't change. And then that nonsense option where we switch out this third base here, and instead of making it, what is that, tyrosine? Sure, doesn't really matter. Right, now we've changed it so it codes for a stop. So we're trying to make this protein, and the whole process just kind of breaks down partway through, right? So we've got these kind of point mutations. But, well, yeah. And then sickle cell was that example we mentioned, right? Where this adenine, right, gets switched out for a thymine. Our glutamic acid becomes valine. And so instead of making this nice, smooth little hemoglobin tetramer that's going to kind of make up our nice little weird donut red blood cell, right, it gets kind of kinked up, right? Because we've switched out, right? Glutamic acid is a polar molecule, right? It's going to behave a certain way. We switched it out for a non-polar hydrophobic amino acid. So this protein is in no way going to fold the same. Right? It gets all hitched up because they've got these weird kinks in them. They get all stuck together and you end up with a misshapen red blood cell by just changing out one nucleotide, right? So the downstream effects can vary greatly from like having no effect to having a really strong set of kind of disease causing effects, basically. Now, So point mutations, single nucleotide things. Other things can happen in these mutations. We can have what are called frame shift mutations. So remember we had a reading frame, right? I've got a start codon. And so if this says start, then the next 3 nucleotides code for some amino acid, the next 3 nucleotides code for some amino acid. But if I add or delete a base in here, right? Say I add a base and it gets shifted down. Now, because all my enzymes and bits don't know any better, right? It's going to read kind of back here. It's going to shift the reading frame, and it's going to basically change every amino acid in the chain all the way down, right? So you can have this shift in the reading frame of a gene. Lastly, before we get into kind of big chromosomal changes, we can have this triplet repeat expansion mutation, which is an absurdly long name for a mutation, but all it means, it basically just says exactly what it is, right? So codons are triplets of nucleotides. That's easy enough. And there are parts of your DNA that get really, really repetitive, right? So like ACG, 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 right? Just like over and over again. And sometimes, right, because kind of the parts in your cell, I don't want to say they're just kind of like dumb little machines, but they kind of are. And sometimes they mess up or they get a little carried away, right? So maybe this little ACG codon is supposed to happen like 5 times, but the DNA synthesis mechanics kind of get a little ahead of themselves. And now you've got like 12 or 15 copies of this little triplet. So we have this triplet repeat expansion mutation where we've got this repetitive part and we just copy it too many times when we copy the DNA, right? This is what causes Huntington's disease and other kinds of neurodegenerative disorders. So the cause of Huntington's, it's not so simple as sickle cell, right? Like we change this nucleotide out for that nucleotide, you get a whole different effect. So there is a repeated part in the gene, in the allele, in this gene, where one of the alleles causes Huntington's disease. So there's supposed to be a repeated triplet. Normally you only have, and it varies. If you start really, really looking at genes, it can get really, really squirrely. There's supposed to be a repeat of this triplet on the order of like 20 to 30 repeats. If you get a mutation where you have more than 30 repeats, you are at risk of Huntington's disease. If you have more than about 45 of these triplet repeats, you will have Huntington's disease. So it's not even, you know, it's kind of one of the scarier things about Huntington's and these other neurodegenerative disorders is that it's not kind of a clean thing. It's not like you have this allele, your child will have this allele. It's like there's kind of this variance in risk depending on how extreme the mutation is. Another odd thing about this triplet repeat expansion mutation is that it only seems to happen, as far as we know, in mice and humans. This is not a problem that we see in DNA of any other organism on the planet. For some reason, something about kind of our enzymes, the machinery in our cells, we, as far as we know, are the only organisms susceptible to this kind of mutation, which is weird. All right, and then these guys. So we've talked about point mutations, single nucleotides. We've talked about kind of some larger effects, frame shifts, right? And then these kind of increasing repeats. You can also have this kind of chromosomal level, right, change in the DNA code. So it can happen a few different ways. And it kind of works the same way as nucleotides, but kind of just at a broader scale, right? So we can have deletion, right? So we've got, say, these different genes coding for whatever, all the way down the chromosome. Sometimes something goes wrong and you lose a whole chunk out of your chromosome. Can happen, right? Not commonly because it's problematic, right? So you can have kind of a whole chunk that gets deleted out of a chromosome. Conversely, you can have a piece that gets duplicated, right? So now you've got kind of a double set of whatever B, C, and D happen to code for, right? This can even happen at the entire chromosome level. Sometimes you end up with whole chromosome or whole genome duplication organisms. Things like spiders have had multiple entire genome duplications. So they've got like hundreds of chromosomes. This can get really squirrely, right? You can have things like inversions. I don't even know how this would happen, but it does, right? So you've got this chunk of a chromosome and somehow in its getting copied, this whole piece gets turned back to front, right? So now you have this whole section of a chromosome that's reading the wrong direction, right? And then lastly, Super crazy, right? So I've got these two chromosomes, say my chromosome 3 and my chromosome 5. It is possible, right? And this should happen with crossing over between homologous chromosomes that are carrying the same genes, but occasionally two chromosomes that are not related, that do not carry the same genes, will trade chunks. So now genes that were previously on my chromosome 3 are found on my chromosome 5. Genes that were on my chromosome 5 are now on my chromosome 3, right? Which is bananas, right? These sorts of things happen. A lot of this stuff is more common in plants. We'll get into that more in chapters 17 and 18. Plants are real weird and don't seem to care what happens to their DNA, but it makes them able to do a lot of kind of cool stuff. They can speciate really fast, all kinds of bizarre things, right? But we can also have these mutations kind of at that chromosome level of change. All right, so all this stuff can happen. So how does that kind of affect like us as a species or other species on the planet, right? These mutations basically are kind of the catalyst for evolutionary change, right? It's how we adapt because our DNA can kind of shuffle and change and modify itself. And so everybody kind of has basically a differential ability for survival, right? These things get passed on. And if they're beneficial, because sometimes mostly they're detrimental, but occasionally they're helpful, right? These mutations then are the starting point for evolution, but you can't have too much, right? There's a whole lot of your DNA that just has to work, right? Things that make your cell divide, things that control metabolism, things that make sure your organs end up where they're supposed to be and do the jobs they're supposed to do, right? So mutation is great and it allows for like variability and all of this fun stuff, but it's got to kind of be within bounds. There's got to be a balance between, right, how fast and how crazy your DNA can change and making sure that your DNA and the things you need to work still work, right? So this balance between variation and just functionality of a species, right? And now, because of just massive increases in computational power, just general ability to extract and analyze and sequence DNA, right? We have genomes for basically thousands of people at this point, or at least DNA sequence for thousands of people. So we can compare that kind of computationally, right? And now we can directly measure mutation rates in humans. And so we've got data on this, which is kind of cool and fun, right? So these various studies have collected this data. So on average, right, every time someone is born, right, these cells come together, these things merge, we make kind of a new set of chromosomes. We've got about 40 to 80 new kind of single point mutation sort of mutations, right? Every individual that's born, right? Just in the mechanics and the odds will be different than their parents, right? Now, that's kind of low stakes mutations. And so what you'll see is kind of the size and severity of the mutation, right? The more severe or larger a mutation is, the less often it's going to occur, which makes sense because the risk then is much greater. And honestly, what frequently happens is if gametes come together and there have been too many mutations, then basically the person doesn't happen, for kind of lack of a better description. But right, we can have kind of insertions of less than 50 base pairs. It can be survivable, it happens, but less often than these little single nucleotide changes. And then those larger chromosomal level things are going to happen even less frequently, right, than these smaller mutations, right? All right, and that's the end of chapter 15. So we won't go through all of these. We'll pick out a few. What is the central dogma of biology? Absolutely. Yep. Information in the cell flows from DNA to RNA to a protein. Correct. All right. And so what is transcription then? DNA to RNA. Yep. DNA to RNA. And then translation is? RNA to protein. Absolutely. All right. So we talked about the genetic code, right? It is a code. It is an information containing molecule, which is kind of a bizarre thing as a concept, honestly. But how does it work? How does the genetic code actually specify amino acids? How does it work? Mm-hmm. Yep. Yeah, so your DNA has a reading frame, right? It basically is a long string of words, right? These little 3 nucleotide codons, right? And every codon codes for some amino acid, right? So it carries that information to the ribosome, which can then make it into a protein through translation. Let's see, what about, what is unique about prokaryotic transcription and translation? Different from how it happens in, say, us. Prokaryotic transcription and translation.
RNA primase occurs in two forms. There's core polymerase and.
So it has, okay, so prokaryotes have one RNA polymerase, yeah, that has, either it has that little kind of sigma factor clamp on the hollow enzyme, or it doesn't, it's just the core enzyme. But kind of more broadly, right, transcription and translation in prokaryotes is different from eukaryotes. How? Not specifically the process. Think about kind of when and where they're happening. Note the Zoom folks are looking at the answer on the screen right now. They're just opting to leave you hanging. Just gonna put that.
Y'all are crazy. Come on now. Shane, I know you got us.
She should. She's just choosing not to.
Or Hannah Calloway, I know you got us too.
They're not having it.
You can flip the script.
Transcription and translation are coupled.
There we go. That was Lexi. I believe that was Lexi. I didn't see.
Lexi.
Yeah, transcription and translation are coupled in prokaryotes, right? They don't have a nucleus or organelles. They're not sending things here or there. It all happens in the cytoplasm at the same time. So DNA is being transcribed, and then ribosomes are hopping right on that mRNA before it ever even lets go to make the proteins, right? So it's coupled. It happens together. What's another one? Ooh, what is the function of tRNA? What does tRNA do? It is transfer RNA, yes. So what is its job? It's made of RNA and it transfers something. Ooh.
For RNA to make protein.
Yeah, tRNA is how is it helping to make proteins? What is it specifically doing?
It's being carried over.
It's carrying what?
The ribosomes.
Yeah, it's carrying amino acids to the ribosome that will be put together into a protein. Transfer RNA transfers amino acids to the ribosome where they get put together. Fab. All right, these are on the slides. These are on Canvas for you to review at your leisure. Let me go ahead. Let's see, because it's more complicated to swap these guys out. All right, so we had chapter 15, which was transcription and translation and central dogma and mutation. So kind of keep all that in your head. We're going to move into our two kind of more applied chapters. So kind of all of these things that we've been covering, what are they good for? What do they get used for? Right, and so chapter 17 is specifically talking about biotechnology. Right, chapter 18 will be more focused on genomics, kind of whole genome kind of research that's going on. But chapter 17 is going to specifically kind of look at these applications of all of these things we've been talking about, kind of building up to for the whole semester. So starting with, not a very compelling section title, but recombinant DNA. It'll make more sense as we get into it. So we're going to talk about kind of biotech generally, and then talk about how, basically how we have become able to manipulate DNA. Right, and that will kind of take us through this chapter. So what is biotech? People use the word biotech either to be like scary or exciting, right? It's one of these words that people kind of tack more emotion onto than is necessary, I feel like. Happens with a lot of these sort of words that like media picks up on. and just uses in silly ways. All biotech is manipulating a biological system to generate some kind of benefit for humans, some kind of product, right? Something to improve the environment, to create medical tools or medicine even, right? To improve agricultural processes, to feed people. Anything that is kind of doing that, right? Using all of these things that we have been talking about to create some product that will benefit kind of humans or the environment.
So in simpler terms, you would say it's used for improvement to create products that help humans grow.
Yep, that'll work. In the end of this chapter, we actually walk through examples of kind of all of these environment, medical, and agricultural applications of this tech. But let's talk about the tech. It's not new. Biotech sounds new. But I mean, we've gotten better at it, but we've been doing it for like millennia, right? We have used yeast to make bread. We have made alcohol since 7000 BC, right? We've been doing it for a while. We domesticated dogs, right? We intentionally, this thing did not exist naturally, obviously, look at it, right? We made them basically over years, right? We couldn't do it with very specific intention, but kind of gradually over time, right? We domesticated dogs, we've domesticated crops, right? Most of the vegetables and things you eat do not look like they looked when we first encountered them in the woods or meadows or wherever it was we found these plants, right? We have changed them intentionally over time to be more like we want them to be, right? And then we started hybridizing things, right? Like hybrid corns and things started happening kind of 1900s at kind of the more global scale when you really start kind of getting crazy with this stuff. But it's not new. We can just be a bit more intentional with the way we're doing it now. And so in the modern era, right, 21st century style, this is pretty much all based around recombinant DNA. And recombinant DNA is just kind of, as its name implies, it's recombined DNA. It's DNA from a couple things that we've combined together, right? A gene that does this and a gene that does this to serve some purpose, right? So we make this DNA molecule from two different sources. We first kind of figured out how to do this in the 70s, initially using viruses and inserting DNA into bacteria using viruses, which is almost kind of like cheating, because if you know anything about viruses and bacteria, viruses live by inserting their DNA into cells. So saying that we got viruses to insert DNA into cells is just like we had the viruses do what they do, one. And then if you know anything about bacteria, right, because they undergo binary fission, we're going to put together lots of chapters at once, right? Bacteria divide by binary fission. We talked about meiosis and sexual reproduction, which allows for genetic diversity. Bacteria get around not doing sexual reproduction by just being able to pick up DNA they basically find laying on the ground and pop it in there and use it, right? So if a virus inserts DNA into a bacteria, the bacteria can technically take that DNA, incorporate it into its own DNA and just run with it. It's a thing that bacteria do naturally. So basically, we just kind of encouraged them to do it in a way we wanted them to. And then later, right, like a year later, we got frog DNA into bacteria, which was a little trickier. Not quite Jurassic Park level, but like on the road to, we're not at Jurassic Park level 50 years later. So, you know. It's, yeah, you hit the can-should wall. We can't quite can yet. They've tried. What was it? Was it a saber tooth? Something extinct that they got enough DNA from, but they can't keep them alive very long. It gets kind of. Oh, the Tasmanian tiger. Was that the one?
Oh, no, I said if you're going to write something on these things, I do see what you said.
I mean, that's fair. I don't make those decisions.
Yeah, really.
We've seen that movie multiple times. It doesn't go well for us.
I know, it's like I want that to happen because I love that, but that's the thing. I think all the death is.
It's like not optimal. All right, so how do we do this? All right, we do this using basically the machinery present in the cells that we're working on already. So as we've kind of covered, Viruses reproduce by kind of taking over other cells' machinery, right? They insert their DNA into something like a bacteria or your cells, right? That's how you get sick by a virus. The virus inserts its DNA into your cells, kind of co-ops it, causes a whole bunch of mess, carries on its viral way. Bacteria can pick up DNA that they find, and bacteria have these restriction endonucleases. So we've talked about nucleases. They're the little enzymes that chop up DNA, right? Endonucleases were the ones that kind of chop DNA in the middle, as opposed to just kind of like clipping nucleotides off the ends, right? Bacteria use these to defend against viruses. So they can have an endonuclease that recognizes a viral sequence. And when that virus shoots its DNA into the bacteria, these enzymes chop the DNA up, right? And they're like, not today, right? So we have restriction endonucleases. We can take kind of all of these parts and kind of use them to our own ends. It's kind of the human story, right? So we discover these endonucleases, we can use them to cut other DNA, right? Because enzymes just do what they're going to do in the presence of the stuff that they want to do it to. So if you take some endonucleases, dump it in a tube with some other DNA, the endonucleases are going to cut it up. It's what they do, right? So we can cut up DNA at certain sequences, because that's the other trick, is each kind of restriction endonuclease looks for a specific sequence and cuts it in a specific place. So now I can cut up DNA at specific places on purpose, which just seems crazy, right? So say I've got some endonuclease. It's going to cut the DNA at this specific point. And it does it in a kind of funky way. So it looks for these, usually they kind of key in on these palindromic pieces of DNA, right? So pieces of DNA that are reading the same forward and backwards. So like, I can't do this off the top of my head, like ACTGCA, ACGTATC. Nope, I can't do it in my head. That's fine. Whatever, it's the same forward and backwards. And so when it cuts it, so say it cuts between the CNA on this end and then the CNA on this end, on the two strands of DNA, right? Because we're a double helix, cuts it on the two strands. So when it's cut and comes apart, you have these little kind of single strand funky tails. We call those sticky ends. Well, now I have kind of uncoupled DNA and I can stick stuff to it if I know the sequence, right? So like this, right? I've got an endonuclease. It's going to cut between G and A in this GAATC, TTC, GAATTC. So I can chop it. When it comes apart, I've got two little sticky ends, these two kind of uncoupled pieces of DNA. Well, I have cut up this other piece of DNA that has some gene that I want using the same endonuclease. It's got the same sticky end. So I just put it in a tube with this DNA with sticky ends. And now I've combined two pieces of DNA, potentially from two different organisms, into one molecule. I've made recombinant DNA just in a tube on a lab bench. It's not that hard, really, actually. So, but how do I know? How do I know what's sticking together? How do I know how big the pieces of DNA? I'm just chopping stuff up. You can't see DNA. I can't like look in the tube and know what the heck is in there. So we got gel electrophoresis. Gel electrophoresis is a way to visualize and sort DNA you've cut up. It also does lots of other things, but for our purposes, we'll stick here. So this is a gel rig. We have them here, over there on that side of the floor, I think. So if you're in the department, if you stick around, you too can run electrophoresis gels. It's not that tricky. So it's just this little plastic container, right, with a place to plug a cathode and anode into. And you make a gel. It's basically, you make it out of agarose gel. It's like really tough jello in a little mold. So you make it in a little mold so that it has these little kind of wells in it. All right, and you put it in this rig and you fill the rig with buffer, right, some kind of ionized liquid that can hold a current, right, because we're going to run a current through it. Then I have this tube of DNA that I've put some nucleases into, so I know my DNA is all chopped up, and say I want some specific 200 base pair piece. Well, how do I pull it? I can't do that by hand. I put it into the gel and say for like half an hour, I run a current, right, through this gel. As the current runs through the buffer and subsequently through the gel will also carry current. It pushes the DNA through the gel. So what? So that the DNA will get sorted by size. So small DNA, or if you kind of think about this, if you were walking through Jell-O, if you are relatively smaller, it's going to be easier to kind of push through the Jell-O. There's going to be relatively less resistance on your person. If you are larger, it's going to be more difficult. You're going to move more slowly through the gel. So say in 30 minutes of running this current through, all your small pieces of DNA are going to be way down the gel. All your big chunks of DNA are still going to be kind of close to the wells, right? And they're going to be spaced out by size, right? And generally you add some kind of like UV or fluorescing dye to them. So then you can like put it under a light and you can literally see where all the different sizes of DNA are. And say I want some 200 base pair fragment. I can go and literally take like a little razor blade or a scalpel and cut that piece of the gel out. Now I have DNA that's all 200 base pairs long, and I know it is, right? So like this, right? Got this DNA, mix it with a bunch of different restriction enzymes. All right, so say I want this 200 base pair piece. All right, well, I've got this reaction in a tube, DNA, and this restriction endonuclease. I put it in to a gel rig, run it for a while, right? The larger piece isn't going to get as far. It's going to be closer to the well. The 200 base pair piece, all of the pieces of DNA that are 200 base pairs long are going to be right here. So I can literally go in, cut this out, and then mix it with something else that I'm interested in, right? So that's gel electrophoresis. Whew. Then, so I can make pieces. I can pick out the pieces I want. Recombinant DNA means that I stick them back together somehow. Scotch tape won't work, so what do? We've got DNA ligase. You can pull DNA ligase out of cells. You remember that guy? Right? DNA ligase is the enzyme that joins strands of DNA, right? When we're copying DNA, it's what stitches all the Okazaki fragments together on the lagging strand. It'll also stick any other pieces of DNA together that need to be stuck together. So I can take my fragments with my matching sticky ends. I can put them in a tube. I can add some DNA ligase enzyme. You can just buy tubes of enzymes if you want to, right? So I can mix them all together. Maybe I want to insert them into little plasmids, little circle DNAs that bacteria will take up. I want to put my little gene or whatever in there with some ligase. I can do that, right? Insert that little gene fragment in there. And then by inserting that historically into bacteria, right? You would put these little plasmids with my inserted gene into bacteria. You would let the bacteria do what bacteria do, which is just multiply all over the place. And they would kind of do the molecular cloning for you. We'll talk about, what do you call it? We've got machines that do it now, basically. But you can do this molecular cloning where I insert a gene into a plasmid and then just make lots of copies of that plasmid and subsequently lots of copies of the gene that I'm interested in, right? So just like this, got this little plasmid guy, this little ring of DNA that some little bacterium won't know any better and will just pick up. I can take one restriction endonuclease, chop a piece out, right? I can go through that whole long process to get this other piece that I'm interested in, this foreign DNA, put it in here, add some ligase, and end up with some amount of plasmids that are carrying the DNA that I just isolated, right? And that's how you mix DNA together. You've made recombinant DNA. You can do it in an afternoon. It's not that hard, weirdly. All right. So we're going to talk about PCR, which is kind of step next, right? Because if I insert something into some DNA, I probably want more than just like one copy of it. I probably want like a whole bunch of it to do something with, right? So how do I make a whole bunch of it? The answer is PCR. So PCR stands for polymerase chain reaction. Right? So polymerase, right? We've talked about polymerases. We're going to be specifically looking at DNA polymerases, so all the way back to, right, synthesizing DNA. And so basically what we've got are these machines, and again, I'm pretty sure we've got a PCR machine here, that I can have a tube of DNA, I can add some enzymes and whatnot to it, put it in the machine, and make thousands of copies of my DNA of interest, right? Because basically what we do is we're going to mimic DNA replication. We're going to add enzymes and adjust temperatures such that we cause DNA replication to happen. We can make millions of copies of a DNA sequence. So we can do this using primers. Remember primers? You can convince polymerase to make copies of things by giving it a primer to attach to, right? So if I have a primer, So I've got DNA. I want some specific piece of this DNA to be copied. I can add primers that are on either end of the DNA I want to be copied. I can have polymerase that are going to copy from the primers, and then just run a cycle of kind of heating and cooling over and over again, specifically Taq polymerase. So we use this polymerase that came from this bacteria, Thermus aquaticus. It's found in geothermal vents. So it's very heat tolerant because we're going to be heating this stuff up pretty warm, right? And so we don't want it to denature because we need it to do its job, right? So we had a polymerase, we have our DNA, we have our primers that are going to mark the piece we want copied. And so we're just going to have this polymerase make copies over and over and over again. So 3 steps. Everything has 3 steps in this whole last unit and a half. So we're going to start PCR by denaturing our DNA, right? We've got a polymerase that is not going to denature in heat, right? We've specifically picked one that's really heat tolerant because we don't want it to fall apart when we heat it up. But we need our DNA to be single-stranded. And it's not going to kind of naturally do it on its own. But if we heat it up, right, that's kind of what molecules do. They kind of unwind and loosen. So if you heat up DNA, right, a double helix of DNA, as it gets warm, it's going to kind of come apart and be single-stranded DNA. So that's denaturation. Now that we've got the single-stranded DNA, we can lower the temperature, and our primers are going to bump into and attach to the spots on the single-stranded DNA where we want to make copies, right? Because they're going to be specific to some piece of the DNA we want a copy of. So we're going to anneal or attach the primers to our single-stranded DNA. And then we kind of take it to this medium temperature, this kind of like enzyme happy temperature, right? So at this Taq polymerase, this DNA polymerase is kind of ideal temperature. We're going to sit there and it's going to find those primers and synthesize the DNA and make copies of the DNA that we want. And so what a PCR machine does is it basically runs through these 3 temperature settings over and over and over again, like dozens of times, right? And so, right. And they basically kind of look like this. You've got this machine, you put your tubes in here, you program in some series of temperatures you want it to run based on your primers and your polymerase, whatever, right? So we denature the DNA, we attach primers, we find a temperature that polymerase likes, it makes its copies, and we just do it again and again and again and again until we get thousands and thousands of copies of the DNA sequence that we want. Yeah. Basically, all kind of using the mechanics of the cell that were already there. We're just kind of doing it like, not in silica, but basically in a little plastic tube. Right? And it usually runs for it depends on the reaction, 45 minutes, couple hours, and then you have a big tube of DNA. All right. Right, because previously, right, kind of what we mentioned initially, DNA had to be cloned into a vector, into bacteria, and then we had to rely on the bacteria to make the copy. So now we got machines that will do what the bacteria was doing for us before, right? And so we would also sequence DNA kind of that way, this kind of step-by-step way. Now we've got this next generation sequencing, which we'll circle back to, uses PCR techniques for sequencing, right? We can kind of make thousands and thousands of copies of DNA. We can have computers reading the DNA and writing down the sequences for us, right? And so this is where the first human genome took 13 years and nearly $3 billion to sequence the first human genome. Today, I could do it for $1,000 in 3 days, right? And beyond that, now we've got these mini-ion sequencers. You can get a thumb drive, this little thing, it's like the size of a thumb drive. I can take a sample of DNA, put it in here, and I can sequence it on my laptop through a USB port. It is bananas what we can do now. It's pretty cool. The reagents for the mini-ion sequencers are still pretty expensive, but you can do it. So we've got people out sampling diversity in rainforests, and they can just bring it back to whatever little tent they're hanging out in, right, and sequence DNA in the jungle. It's pretty cool. All right. So what can we use all this for? That's for Friday. So we're going to talk about how we use all these tools to create, correct, and analyze genetic code. All right. And then we'll get, I think, into some more applications. So that's enough for today. Have a good rest of your Wednesday. We'll pick this up on Friday. Oh, there were some missing lecture journals.